I am Muralidharan Bashyam. I'm from Center for DNA Fingerprinting and Diagnostics, which was nicely introduced by Dr. Kasbekar. And I work mainly on cancer, like he mentioned, and we work on 4 or 5 different cancers which are part of our orodigestive tract. And I also do some work on genetic disorders, which I'm not going to talk about to you today. But firstly, I'd like to thank the Academy and Dr. Kasbekar for giving me an opportunity to talk here. This is my first meeting, attending the first editorial board meeting, and also first time I'm giving a talk in association with the Academy. It is difficult to follow somebody who is endowed with expertise in Tharuri and English. And genetically, I'm supposed to be endowed with this because I'm a South Indian Brahmin. But I'm an example of epigenetic scoring over genetics. I was born and brought up in Delhi. Hindi is my first language. Second language is Punjabi. Third is Haryanvi. And fourth is Tamil. And although genetically I'm a Tamilian, homozygous. Both parents are Tamil Brahmins. But I think in Hindi, I eat Punjabi food. And I have to translate every Hindi sentence into English, although I've been doing this for 25 years. Yeah. So that's an example of epigenetic scoring over genetics. Okay, so this is just to introduce the colon and the rectum, which constitute the large intestine, a very important part of the digestive tract. The major function of the large intestine is to absorb water from the stool. Okay, all the other absorption is mostly done in the small intestine, which is this part, and it's actually much longer than the large intestine, but it's kind of, imagine it like DNA being convoluted and occupying a much smaller space than the large intestine. So all the nutrient absorption, or most of it, happens in the small intestine. Large intestine is only absorbing water mainly, and basically if it doesn't do that, you have loose motion. Okay, this is the ascending colon. This is called the transverse colon. This is the descending colon. This is called a sigmoidal region because of its shape, and this is the rectum, and then the terminal part is the anus. Okay, so let's look at colorectal cancer, what is known worldwide. So it was till about a decade ago, it was classified as a disease of the Caucasians or the white population or of the Western world, disease associated with lifestyle, with urbanization. And in India, it was pretty rare. And in the US or in the Europe, colorectal cancer is mainly defined by an aging population, and most of the cancers arise in the colon, which is the first, let's say 80% of your large intestine, the ascending, transverse, and the descending colon. Very few cases occur in the rectum. So when we started this study in India about 18 years ago, 2004, 2005, we were approached by the clinicians actually. So this project initiated from the clinicians, not from a research laboratory. And they told me that in Hyderabad, at least, they said we see a lot of rectal cancer cases, and pathologically they look slightly different from what we see from colon and what we have learned from the literature. So imagine most of the medical education or medical practices in India are still defined by the British and to some extent from the US medical literature, where everything is defined on a white Caucasian male, if I can use that term. We know it's an insignificant fraction of the world's population, but for some reason everything is defined by the white Caucasian male. Okay, so what the clinicians told us, we have a lot of patients who are young, below 50 years of age, not seen in the Caucasians. We have a lot of patients who have rectal cancer, again not seen in the Caucasians. Our pathologists tell me they are different, but we have no idea. Can you do genetic profiling? And they went back, at that time, like imagine 2004, 2005, the clinicians actually told me what pathways drive colorectal cancer, and I was not aware at that time. And then I had to go to the literature and read about those pathways so that I could apply experiments which can define those pathways and see, are they same in rectal cancer occurring in young individuals, or are they different? Okay, and during this time we realized that even in US now, last 10 years, there's a transition. Rectal cancer is going high, and colon cancer is kind of contained, but the increase in incidence in the last decade or so in Caucasians is actually contributed largely by rectal cancer, which is kind of a traditional problem of our own. Okay, so this study began with PhD student Ratish and followed up by another graduate student Raju Kumar. Both of them have left. Ashmala has also left. She worked on familial colorectal cancer, which I'm not going to describe to you. And Kalyan helped all the students because he was the one who I could train in molecular pathology, looking at tumor sections, doing immunohistochemistry, doing hematoxylin and eosin staining, and running to the pathologist, and sometimes being a pathologist on his own. And he's still there in CDFD, but he's part of a central facility now. So this is just to show, in the beginning we saw that incidence of rectal cancer was high across all age groups in India, but it was significantly higher in the younger population if you look at the rectal cancer fraction. And in fact, so much so that the global cancer report, the latest one, actually shows India is third in the world in terms of early onset rectal cancer incidence as well as the death rate. So in fact, for the death, it's second, tops US as well. So this is what we reported when we started the study, that this particular cancer, which we call early onset sporadic rectal cancer, is the predominant CRC subtype in India. And we went ahead and looked at, like I mentioned, the pathways which drive colorectal cancer. So what is known again from white Caucasians is that majority of colorectal tumors are driven by a signaling pathway called Wnt signaling. This is very well defined in the literature for almost 3 decades. And the main molecule which is like the target of the Wnt signaling is a protein called beta-catenin, and its major job is to anchor the E-cadherin to actin cytoskeleton. So E-cadherin is an epithelial glue which holds different epithelial cells to each other. Okay, and how the epithelial cell is anchored to the cytoplasm is through beta-catenin, and there are alpha and gamma-catenin also, 3 different proteins, and they anchor E-cadherin to the actin cytoskeleton so that they are fixed at their location. So the default pathway where Wnt is off, that means there is no signaling, the free beta-catenin which is in the cytoplasm is degraded by a cytoplasmic destruction complex, where one of the most important proteins is APC. It's almost 2700 amino acid long protein. It's a major scaffold of the destruction complex, and the complex has 2 kinases which phosphorylate beta-catenin and mark it for degradation by the classical proteasome degradation pathway. But when Wnt signaling is on, so you have a Wnt ligand binds to its cognate receptors, this binding disrupts the complex. So it pulls some of the components here, the other components are not active. So the free beta-catenin, which is not binding to E-cadherin, can now enter the nucleus, and there it acts as a transcription factor, activating genes involved in cancer like cell growth as well as cell invasion and metastasis. So kind of all aspects of cancer is driven by beta-catenin, which cooperates with classical transcription factor families of TCF and LEF. Okay, so this is what was known, and all the literature pointed this is the major pathway driving colorectal cancer. But again, when we say colorectal cancer, it is colon cancer in individuals aged above 60 or 65 years. Okay, so this is the APC, and it has several different domains. It's supposed to be a multifunctional protein, but for colorectal cancer biologists, the importance is that most of the cancers arise due to inactivation of APC. So you don't need the Wnt growth factor to bind. In absence of Wnt signaling from the membrane, APC is already active or rather inactive because of the mutation. So the destruction complex destabilizes, and beta-catenin is stabilized and can enter the nucleus. And most of the mutations occur in a region called mutation cluster region, and these arrows actually define the major beta-catenin binding domains. And all the mutations in colon cancer are protein truncating mutations. So this is the region where beta-catenin binds and is degraded, but most of the mutations, like I said, truncate the protein. So beta-catenin cannot bind here, and the scaffold also is disrupted, and the protein can now enter the nucleus, and there is an aberrant or a constitutive activation of downstream targets by beta-catenin. Okay, so very simple, and everybody believed this was true. We also believed this was true. And you do a simple immunohistochemistry on your tumor samples. So you see beta-catenin in the membrane. You say, okay, there is no Wnt signaling in these tumors, and you have colorectal tumors where beta-catenin is sitting in the nucleus, and you say that this particular tumor is Wnt positive. So a simple immunohistochemistry test, few hundred rupees, you can define your cancer whether it's Wnt positive or Wnt negative. Okay, so we did lot of work on colorectal cancer, which I'm not talking to you about today. Everything is published. What I'm talking to you is actually not published yet. And one of the things we did was this particular subtype, early onset sporadic rectal cancer, we did next generation sequencing based exome sequencing. Exome sequencing meaning you're sequencing all the exonic region of the genome, so roughly 2% of the genome, to find out if there are coding region mutations which could be driver mutations. The idea was, can we pick up something which is not known in colon cancer, because there we know it is APC, TP53, KRAS, and PIK3CA and some other known protein. So we did pick TP53. APC mutation was less, the frequency was much less than what you find in Caucasians. This is a worldwide study, but mainly representing Caucasians from US. And KRAS mutation frequency was kind of similar, maybe little bit less. Okay, but we thought, okay, APC mutation is less, and maybe there are other drivers of cancer, and which we actually found in this manuscript. It is one of the drivers is ERID2, which is part of a SWI/SNF chromatin remodeling complex. But we were stuck with this APC frequency, and we thought, okay, let's see the Wnt signaling status. Let's look at the beta-catenin status. What is happening in the tumors where APC mutation is there? Okay, so we were surprised. So again, work done by Pratyusha, and now being followed up by Sanjana, and Padmavati is the technical help in the lab, kind of she replaced Vishwa Kalyan. So we found 22 samples which had APC mutation. Some were biallelic, most of them were monoallelic, and less than 50% had beta-catenin in the nucleus based on the simple immunohistochemistry test. Although we have been told by everyone at all the leading conferences that if you see APC mutation, beta-catenin is in the nucleus. So we went to the literature, and again, we had done this before, but we did it again. And surprisingly, people reported APC mutation. They've been reporting for last 20 years, but nobody did an immunohistochemistry on the tumor samples to look at beta-catenin. They just believed what they were told was true. And only some reports from a familial form of colorectal cancer, which is called familial adenomatous polyposis, which also has APC mutation driven cancer. Only difference being there it is a germline mutation. There were reports people were doing immunohistochemistry, but there was very good concordance. You have APC mutation, you see beta-catenin in the nucleus. So as usual, I mean, as scientists, we always suspect our results. So I said, no, this is something wrong. Let's look at everything. Maybe there's some labeling error. The samples are not matching with what the sample used for mutation is different from what you did in immunochemistry and all that. So we looked at other scenarios, like what if the fraction of tumor cells that have the mutation is very less. So because next generation sequencing is of high depth, you're bound to get even low frequency mutation. So we looked at the reads, the fraction of reads which have the mutation, which gives you a estimate of what fraction of tumor cells actually have the mutation. So remember, tumors are bulk, and we take 1 or 2 centimeter diameter tumor piece, and we just mince it and isolate DNA. You don't know from which part of the tumor you're getting the mutation. So when we looked at the fraction of tumor cells having the mutation, whether beta-catenin is in the nucleus or not, the fraction was kind of similar. In fact, most of the cells, so imagine being a heterozygous mutation, in most of the cases you expect it to be around 0.5, and if it's homozygous, you may get more than 0.5. Okay, so so this was not the case. The fraction of epithelium having mutation was same whether beta-catenin is there in the nucleus or not. What about KRAS? So there were reports which showed KRAS mutation can drive beta-catenin to the nucleus. So that APC mutation may not be sufficient. You need an oncogenic activation of KRAS as well. But again, KRAS mutation status not different in the two sets of samples. If anything, it was slightly higher in the nuclear negative, but definitely no correlation. Okay, so next we thought we look at the position of the truncation. So what was known in the literature was that a biallelic loss of APC is kind of lethal. The intestinal epithelium cannot survive if both copies of APC are lost, because there is too much of beta catenin in the nucleus and the cell kind of commits suicide. It cannot withstand so much of transcriptional activation by beta catenin. So most of the mutations or truncations still leave one or two or three of the domains, so that the binding of beta catenin is still kind of rudimentary and not too much of beta catenin enters the nucleus. So this is the knowledge from the literature. So we thought maybe that is the difference between the nuclear positive and nuclear negative. How many beta catenin binding domains are left in the truncated protein? But again, when we compared the number of beta catenin binding domains left in the samples where beta catenin was found in the nucleus or not found in the nucleus, no difference. In fact, two of the samples where beta catenin was in the nucleus, the truncations, we had full length APC basically. So the protein was truncated, so this is slightly incorrect, but it was truncated beyond the beta catenin binding domain somewhere here. So no difference here as well. Okay. So we were stumped. Then we went and answered the question which I posed to you in the beginning. What if the immunohistochemistry represented a different area of the tumor and the mutation came from a different area of the tumor? Okay, this is entirely possible because we mince everything, isolate DNA, and we take a section from somewhere and do the immunohistochemistry. Okay, so we went to the paraffin block, which is how a pathologist stores tumor samples, and these are formalin fixed paraffin embedded tumor samples, and this can be stored for years, nothing happens. Because of the fixation, the DNA RNA quality is compromised to a large extent, but still you can do immunohistochemistry or some of the other things. Fortunately, we have been working with paraffin labs for a long time. We had standardized techniques to isolate DNA and do mutation sequencing, okay, just PCR based Sanger sequencing. So we isolated tumor cells from the section and screened for APC mutation, which we knew was supposed to be there in this sample. And we also did immunohistochemistry for the whole section to see if this kind of matches. And actually that's what we found. So here are examples from 3 different tumors where beta catenin was in the cytoplasm. So we repeated the immunohistochemistry, and from the same cells we isolated DNA and confirmed the mutation which was found from the next generation sequencing approach. So again, actual cells, epithelial cells, tumor epithelial cells where beta catenin is not in the nucleus, you have classical APC mutations which are supposed to drive beta catenin to the nucleus. Okay, so again, not answering our question, and this is as a control. We took a sample where beta catenin was in the nucleus, and there, yes, we did find the mutation from the same cells. Okay, so we did several of these. I'm showing you 3, and again, we can't explain why or what is going on. Finally, we went to cell lines. So cell lines are a more defined system, and we looked at colorectal cancer cell lines where APC mutations were already reported. Some of them had done immunofluorescence, but we just wanted to repeat it. So we repeated the sequencing from 6. I'm sorry, this is kind of. Yeah, still. So basically, we had 7 cell lines, colorectal cancer cell lines, where APC mutation was already known, and we sequenced and confirmed those mutations. 3 of them we found beta catenin in the nucleus. This is an immunofluorescence experiment, not immunohistochemistry. And the KRAS mutation status, again, not correlating. These are 4 cell lines where beta catenin is in the membrane. And again, there are different mutations. These 2 have biallelic mutations. One of the allele has all domains intact, the other allele has only 2 domains. Again, KRAS wild type or mutant, doesn't matter. And this is one cell line where wild type APC, beta catenin was in the nucleus, and we found the mutation. So not only what we found was true from tumor samples, okay, but it also, we are finding the same thing in cell lines, okay. So yeah, I would kind of end here. So I'm sorry, I'm not going to answer the question that we started with, but it's a question that we are posing to the Western literature, that this is what was reported, but we are not finding it. And maybe others found it, but nobody reported. So we have put this up in a preprint server called bioarchives where everybody can look at it, and we are trying to publish it now. Hopefully some journal will take it. So what I've shown you today is that after 3 decades of Wnt beta-catenin research, we thought you have Wnt signaling or you have APC mutation, beta-catenin goes to the nucleus. Those studies are mostly based on cell lines, very few studies on actual tumor samples. So we find APC mutation may not correlate. So maybe there are additional cellular signals which are needed to drive beta-catenin nuclear localization. We thought of KRAS, which others had suggested, but maybe there is something else. And what is more important for us is there a non-canonical role for mutant APC? So remember, large fraction of tumor epithelium have APC mutation, which also means it's an early event because you find it in almost every tumor cells in your tumor samples. So it is classified as a driver event. If it's not activating Wnt signaling, it must be doing something else very important for the tumor. That's why the tumor has selected and retained this event in almost all epithelial cells. And there are some reports again in the literature, some talk about, remember it's a very large protein, there are other domains, so there are domains which are known to bind to microtubules, and if that is lost, there may be perturbation during metaphase to anaphase transition leading to chromosomal aberration. So those are not very well defined studies, but there are literature showing that and some other functions for APC not related to beta-catenin signaling. So yeah, finally I'd like to thank all the hospitals from where we get excellent support, patient clinical data, pathologists, surgeons. Saverinathan from NCBS, he's our collaborator for analysis of next generation sequencing. Most important, all the patients who consent to be a part of these studies. And these are the funding agencies which support most of our work related to colorectal cancer. And I thank you all for your attention. So, yeah, I don't know how we are doing for time. Okay, so we can have questions now or during the break? Yeah.
A very interesting piece of work. So you expect these APC mutations to originate in a somatic manner and not really in germline?
Both are possible and are known. So if you have a germline mutation, it's usually monoallelic, or almost always monoallelic. During the course of the colonic epithelium division, you may get a somatic hit in the second allele, and then you get full-blown cancer in these individuals. That's called familial adenomatous polyposis. It's a syndrome which causes familial colorectal cancer. But that is very less, like 2 or 3% of all colorectal cancers. Majority are sporadic, and you're supposed to have a biallelic event. So you can have one mutation in the APC, truncation mutation. The other allele can be lost or can be methylated, like silencing by methylation, or mostly these two, or you can have a biallelic mutation. But yeah, mostly somatic, definitely somatic, yeah, because germline is very rare. Yeah, so both are known, yes.
And both those copies need to be truncated in order to show this phenotype.
Yeah, so unfortunately, we didn't have enough material to look at loss of heterozygosity, but we have indirect evidence because, like I said, you have very good fraction of tumor epithelium, and you won't get that if the other allele is lost. So we think both alleles are there, and but we're definitely not sure about LOH. We'll have to do that. At least some samples we have enough material to look at, and methylation also we have not looked at methylation. But one again indirect ways to look at the transcript. But again, we would like to do it from the sections rather than the bulk tumor. But transcript is more difficult than mutation screening from the paraffin sections. But we are going to try that. Yeah.
So I was interpreting the dissonance of the APC mutation and mobility to the nucleus. Wnt has a lot of non-canonical functions, which have very different outputs.
Yes.
So could there be any correlation of, because this is a canonical,
correct,
probably for the students also.
Yeah.
You can say this, if I can say that, Wnt has 2 lives or multiple lives. Canonical means the main life and a lot of side life. So as Dr. Bashyam showed that even if mutation is there, Wnt does not go, which it normally should. And my question is whether non-canonical functions, which is related to calcium signaling and whole bunch of things,
Yeah,
continue.
so the answer lies in the thesis dissertation of Rajakumar, who submitted 2 or 3 years back. So he did transcriptome profiling of Wnt positive samples versus Wnt negative samples. Again, same early onset sporadic rectal cancers tumors where beta-catenin was in the nucleus or not in the nucleus. And we did not actually go back and look at the APC mutation status really closely, but what he found was the enrichment of calcium N-fat signaling in the tumors where beta-catenin was not in the nucleus. And of course, we had both APC mutant and non-mutant samples in that category. But so we proposed that calcium N-fat signaling, which actually is also a non-canonical arm of Wnt, which is unrelated to beta-catenin. And we did find increase in expression of some non-Wnt ligands. Okay, so there are about 19 or 20 Wnt ligands known in our genome, and all of them are expressed, not together, but some of them will bind to their cognate receptors, which I showed, and trigger beta-catenin stabilization. So frizzled and LRP. There are some orphan receptors where other Wnt binds, and we found increased presence of those receptors, the transcript of those receptors, as well as those Wnt ligands in these samples where beta-catenin was not in the nucleus. So the short answer to that question is yes, we do see non-canonical Wnt, but that's it. We kind of not yet gone beyond and seen which, because there are 2 or 3 different non-canonical Wnt. We know it's calcium in this case, but what actually triggers which ligand and is there a separate mutation and all that we have not seen.
There's some clues to that, because yeah, we can continue.
Yeah.
But yeah,
In fact, students can ask questions.
yeah, I think
If any students have any questions, then we go for tea and they can
Sure.
more informally speak with and anybody else. Maybe, you know, that go in this sense.
Just out of curiosity, could this be because of this genetic background effects? That's something worth looking at. The fact that you don't get this beta-catenin localization as you expect in patients with APC mutations.
Yeah.
Maybe it has been reported in populations where you have other types of genetic backgrounds which cause this correlation.
So yeah, there are reports from Asia where they don't see where the frequency of APC mutation is less, but they have not looked at beta-catenin localization status in those tumors. But yes, there are reports from Middle East, from Malaysia, and also from the Oriental world that the APC mutation frequency is less than what is reported from the Western literature, and it's close to our frequency, 40 to 50%. That has been reported. Yeah.